Thanks so much, everybody. It's a pleasure to be here today, and thanks so much for the organizers to invite me and tell you a little bit about what we're doing at the Jackson Laboratory and how mouse models can really help and influence a number of different rare diseases, but especially ALS. I'm a neuroscientist by training. I have worked in mouse genetics for about 30 years, and the majority of my career has been spent at the Jackson Laboratory. And if you're familiar at all with the Jackson Laboratory, you know that if you really want to model human disease in mice, this is really the place, to do it. The facilities, the access, the resources. There's just really no better place to do this. And I've also had, the distinct opportunities at JAX to not only do my research there, but to start big programs as well. And especially in areas where, there is an unmet need. And then quite frankly, I think where we just need as a community to sort of stand back and look at the way we do things. And, somebody said to me once, if you don't change the way you look at things, the things you look at will never change. And having been at a place, for as long as I have, it was really easy for me sometimes to sort of step back and think about where we need to make process change and where we can make improvements. Because at the end of the day, academic research is great, and I love industry and biotech, but there's a sense of urgency for our patients that we need to address in a more immediate way. So the ALS resources at the Jackson Lab were really, I'll also say that I lost my mom to ALS about 15 years ago. So this became really personal to me, I think, like it is to many people who have an experience like this. And you step back and you look and you say, well, what are we doing? Where are the mouse models? Where are our resources? Why are they scattered? Why aren't they centralized? Why are we taking material transfer agreements? Why is it taking so long to get the resources into a centralized place? And so with that, I really took, some lessons from the spinal muscular atrophy area that I worked in for many, many years, who very successfully, pulled their resources and their community together and have successful therapeutics at this point. And so, really the ALS resources, I've been very fortunate to work with so many talented scientists and clinicians around the world and have convinced them, I think, for the most part, that we really need to centralize our resources. If we don't centralize our resources, access to these mouse models in order to test therapeutics will happen much more slowly, and we need to really accelerate that. So our mission was really to work with the scientific community to provide these rapid access to mouse models, and then also to do drug efficacy testing as well, because it's not necessarily the easiest thing to do. And when you think about the technology advances, there's a lot of people whose technology is fascinating, but they've never seen or touched a mouse before in their lives. And having for them to go through the learning curve of something we do every day, like brushing our teeth, was just didn't seem reasonable to us. And so a lot of people ask why the mouse. And so for some of us, I think this is obvious, but for others, we have heard a lot about iPSCs today, and we know there's the cellular models that are fantastic, Drosophila models, C. elegans. There's a number of different model organisms that really, are very helpful in studying various diseases and provide, rapid access to some of the questions that we want to answer. But at the end of the day, we need a mammal. We need a mammalian model that demonstrates the same sort of biology and physiology so that we can study disease mechanism and pathophysiology in a mammalian system. Mice have an incredible conservation of gene, protein, and pathways, and so the biology is really conserved from mouse to humans. And mice are just really, from the standpoint of a mammalian system, relatively inexpensive compared to things like non-human primates, relatively accessible, and able to answer a lot of the questions that we need. And then for drug testing, and this is something having been aware of more so in the rare disease community, where efficacy is not necessarily going to be demonstrated that readily in clinical trials. I think the aspect of having a mouse model that really does show you efficacy really de-risks this for companies and for industry. And it also convinces ourselves that, therapeutics that we're going to put in patients, are necessarily going to be not rolling the dice, but really do have a chance at benefit because we've already demonstrated this in the mouse models that we have. So the problem is that ALS is a very complex disease, as we've just heard and we all know. And I think the problem with it is the heterogeneity within the disease population. And the other component of it is that, you know, most of ALS is not inherited. It is sporadic. But I will say that there are certain convergent technologies and therapeutics that have really, you know, sort of, I think, spurred the scientific community, especially myself, you know, to really think about, you know, ways that we can do things bigger, better, faster, stronger, and let's just move towards that. And one of them has been diagnostic sequencing. And so, you know, what I'd like to try to convince you is that in some cases, in a very complex disease, you can take the rare forms of the genetic mutations and go from rare to common. And moving from rare diseases where we know so much about the genetics of the rare populations, even though they're only 10%, are really going to inform us about the biology, the pathophysiology, the disease mechanisms. And I think for things like, for example, like TDP43, where we know the majority of ALS converges on TDP43 pathology, having a mouse model that's genetic is only going to help us inform the sporadic forms, you know, in the patient populations. And so, so this is a diagram. I think you've seen a version of it already. And I'll say it wasn't long ago in my career where SOD1 G93A was the only mouse model that we had for ALS. And it's unfortunate because I think that what we know about SOD1 G93A is that it really represents a very small portion of the patient population, and its disease mechanisms are somewhat, you know, vastly different from the majority of the other cases of ALS. But nonetheless, as science has progressed and genome sequencing, you know, has really provided us with an opportunity to not only identify those genes that are causative, but also that are risk factors, we've been able to now take a lot of the genes and convert those into mouse models. And I was very fortunate, again, to be at the Jackson Laboratory, where not only can we study human disease, but we can genetically engineer anything you want 9 ways till Tuesday, and really, really quickly. And so when we started understanding what these genetic mutations were, we started engineering them ourselves, not because we were going to study every single one of these, is because we wanted to put them in a united or a unified repository where anybody could have access to those models. So nobody else has to bother with engineering them. We already did it. Nobody has to bother with how do I get access to it. We already have it. And so, you know, these are areas where with profilin, C9orf72, TDP43, and I've been fortunate to work with a number of individuals in this room, you know, to really provide a think tank of what are the mouse models that we need and how can we use them. So if you go to the Jackson Laboratory, you'll see this, our website and our catalog. We do have one of the largest repositories for mouse models for ALS. And of course, we work in a number of different areas as well. But ALS has really been a disease area and focus, you know, that we've really tried to concentrate on, and getting these resources all together. And if we needed to genetically engineer them in order to make them accessible, then that's what we can do. And I think that what I like most about this, and this was a lesson I learned from spinal muscular atrophy, is that access to these mouse models, you know, are really critical for individuals, and having access to the same mouse models with the genetic quality control and all of the things that you need, so that if you want a mouse model, you know, for a particular, you know, scientific experiment, all you have to do is call and order it. I mean, it's just that simple. There's no MTAs, and for the most part, we try to reduce the burdens of licensing whenever we can to convince tech transfer offices to make this more accessible. So the other thing that we decided to do, having worked with spinal muscular atrophy for so many years, is access to the mouse models were great. But when you really have worked with the mouse models and you know them probably better than anybody else, it's a lot easier to just work with companies and academic institutions who have a therapeutic that they want to test, or a hypothesis that they want to look in. And so in addition to making these mouse models available, you know, we started this in vivo pharmacology center here at JAX. And so this is where, you know, again, if you're not necessarily working with the SOD1 model, There's no reason to order it. There's no reason to breed it 2 by 2. There's no reason to take 6 months in order to get a cohort of mice. And there's no reason at all that you should have to spend, so many years, trying to understand the disease pathophysiology of that mouse model when we can just simply do that for you if that's the best way, or work with you to help you understand this. So most of our studies are designed for prophylactic and therapeutic. If we have a drug that people want to test for us, we usually end up going in sooner rather than later just to see what the maximum efficacy could be. And then we test in the course of the disease as well. We have mouse models for SOD1, profilin, C9orf72, which is the most common form of inherited ALS. And we're not biased to the models that we make here at JAX. Whatever animal model you want to test something on, we're happy to do it as long as we have the model at JAX. And so looking at things like cytoplasmic aggregation of TDP43, making sure that we have those assays, the antibodies, everything together, we are hoping, and I think it does, just accelerate how quickly we can investigate potential therapeutics. And so this is really how we're formed in terms of the in vivo pharmacology testing center. We work a lot with rare diseases, especially because from an academic and industry standpoint, we were never really designed to help N of ones. So we have a very big emphasis on the ultra rare diseases as well. But the common rare diseases like ALS, Friedreich's ataxia, Duchenne muscular dystrophy are really areas where we have a lot of knowledge and excel, and not in addition just to the therapeutic testing, but to the biomarker cores as well. So looking at things like neurofilament light chain, looking at them longitudinally and how that works. And I have no idea how I'm doing on time. Oh, I do. Well, that's great. Wow. Okay, so so how does it look when you're going from mouse models to what we see in patients? And so this is an example, and I will say that mouse models don't 100% translate. We know mice aren't people and people aren't mice, but there are certain things that we can investigate and we can do it relatively rapidly. And these are areas again where we can de-risk certain therapeutics. We don't necessarily want to waste time in a clinical trial where we don't have any evidence whatsoever that the drug is going to be efficacious. And so understanding and maximizing our knowledge by using mammalian mouse models is really a relatively good strategy. And of course, the FDA really wants to see this data. In addition to the safety, they want to see efficacy. Will you be improving the quality of your patient's life in this particular instance? And so this is a SOD1 mouse model that we use. And so we try to concentrate in all of our areas in the pharmacology group to look at the outcome efficacy measures that are going to be most important and most informative in the patient trials. And so there are things like rotor rod and grip strength that we look, but we also look to see what we're going to use in the clinic and see how we can model those particular outcome efficacy measures in the mice. And so here we're using EMG and compound muscle action potential. We use the recording from the hind leg, and over again, we've established this clinical outcome measure in the SOD1 G93A mouse, as well as other mouse models for ALS, and really looking at that deterioration of muscle amplitude, and also decrements and repetitive nerve stimulation. And that's just continuously stimulating the muscle in the nerve in the mouse and looking for the muscle to fatigue faster. And so in these mouse models, we see a certain amount of motor neuron degeneration, axonal degeneration, all of which is very measurable in a longitudinal fashion. And from a histological standpoint, we also have outcome measures as well that can help us really look at how we can translate what we see in the mouse to the clinic. And so we like to try to do things in a very efficient way. So the mouse models that we enroll will be in a particular time that are usually just prior to disease onset. We can baseline the compound muscle action potential and the repetitive nerve stimulation as well as the rotor rod. And these are really predictable in the mouse models over and over again. And so the variability that you might see from lab to lab, depending on the techniques and also the personnel, in addition to the health status, is something that we have controlled for extremely well. And so we can take these particular mouse models, we can introduce a therapeutic. Again, we can introduce it prior to disease onset, or we can put it, introduce it during disease onset, because that's really what we're going to see in the patient population. But in a lot of times, it kind of gives you a little bit of a hint. If you don't see anything prophylactically, you're probably not going to see anything with respect to efficacy during the disease onset. And so we have certain strategies that we like to work with and guide people to when they're doing these mouse models. And I think the other thing to really point out is that, and it's really too bad because I keep seeing them, I review papers, I review grants. The studies are just not powered high enough. And this is something that we've been riddled with in the scientific community over and over again. Because people will use 5 or 6 mice and they'll see an extension of life, and we already know that power analysis is nothing more than an anomaly. And so really trying to educate the scientific community on what a really well-powered study should look like, and how we should move forward with it, I think is really important, because not everybody really appreciates the number of mice, especially in a particular disease model that does have some variability associated with it. If you're going to invest the time and effort into doing the experiment, you might as well do it right, and figure out exactly the number of animals that you need. And so this is just an example, from the time, and so you can really get an idea, starting at 7 weeks, just a few weeks later, whether or not your drug, is going to have an impact in this particular mouse model. And so just to kind of give you an idea of, how a SOD1G93A mouse model, looks like. This is their compound muscle action potential. And you can see at around 55 days of age, they're starting to lose. There's a deficit here that becomes very prominent from the wild type animals. And we can follow this longitudinally. So if you start dosing, let's just say at 40 days of age, what you're really going to want to see is an improvement over vehicle treated animals, that the compound muscle action potential, has been preserved, and the integrity of that is clear. The same is true for repetitive nerve stimulation. And again, this is a survival plot for SOD1G93A, but you can set your watch to it with this model because we've used it for so long and so diligently, exactly when CMAP and repetitive nerve stimulation deficits are going to appear, when we're going to see deficits on rotarod, running wheels, and of course, axonal counts as well. And so, not to be deterred, and also, to really take advantage of all of the different mouse models that have been, available to us into the scientific community. We've worked with not only SOD1, which has been the industry standard, but I will say it's been the industry standard because that's all we had for so long. I mean, we just didn't know the genetics behind ALS, and now that we do, I think we're really making a race to make sure that we have these mouse models. And so there's a few that we use that we find are reliable and informative to some extent. We have mouse models for TDP43, one with the nuclear localization signal that Virginia Lee made. And so when we're looking at TDP43 aggregation in the cytoplasm, and we're looking to see, how we can disrupt TDP43 or alleviate TDP43 aggregation, this is a great model, and we've used it over and over again. The same is true for the SING model. There's some criticisms that it's a little bit, this is a transgenic animal with I1 promoter. It's a little aggressive in its disease course. And so like the SOD1G93A, it's also very difficult to intercede when the disease is really aggressive and on its course. But we feel like this is one where we're looking to not make sure that the animal is alive at 50 or 60 days, but we just want to move the needle a little bit when it comes to survival and see if we can, gain any benefit with that. We've also worked a lot with profilin mice. We've generated some ourselves. And have worked very closely with John Landers. This is another genetic model for ALS. And there are even more that I'm not going to talk about today, but I'm going to try to hit the highlights, at least for the ones that we've reduced to practice in terms of having a therapeutic platform. And I think the other one is C9orf72. This is the most common form of inherited ALS. It's caused by a repeat expansion. It's not necessarily easy to model repeat expansions in mice because they don't like them. And when you try to genetically engineer them, they contract their repeat expansions. But we do have a few. One that I made with Bob Balo. And this one is one that was generated in our lab and studied by Bob at Cedars-Sinai. We have one from Laura Raynham's lab, and then we have an AAV9 from Len Petrocelli's lab, where it's just a repeat expansion packaged in a viral vector that you do an ICV injection at P0, and you get some really nice pathology in the brain that looks very, very nice to the DPR pathologies. And so we just heard a really nice talk on DPR, pathology and how to intervene. And this isn't just one of many mouse models. And we have a few more with target ALS that we're working on so that we can have mouse models that maybe don't have so many tandem repeats and also are completely license free for better access to individuals. And so you can tell me when I'm getting close. 9 minutes. Okay, great. So the AAV9 is a really great, platform that we've used, and this is from Len Petrocelli. And, again, I'll kind of highlight the, components of this. It's a packaged, G4C2 repeat expansion into a viral vector that you do an ICV injection on at P0. Not many people know how to do a P0 injection, into the ventricles of a mouse. But not only can we do an ICV, but we can do intrathecal, we can do ICM. Any route of administration is just really very, very easy for us to do. And so again, we're trying to lend our experience and the benefit of our technical expertise to the rest of the scientific community. And one of the reasons why I probably neglected to say this, that we do it as a fee for service, is because I don't want to fight with you about your intellectual property. I don't care. Right. Your intellectual property is your intellectual property. And I really don't want a whole bunch of lawyers redlining agreements, for sponsored research. It's just so much easier to have a master service agreement where we say this is fee for service, you pay us to run the experiment, and all the data belongs to you. It's quick and it's easy, and there's, no fuss, no muss, and we just go about, getting the work done. So in this particular model, we have really great neurodegeneration, we have protein pathology, we have motor and cognitive dysfunction, and we also have a lot of biomarker assays. I'm not going to run through all the data for you, but this was really generated and worked in Len Petrocelli's lab. He did a fantastic job. He's given us the virus to use. Mayo is, we licensed the viral vector from Mayo. And we validated, all of these particular outcome measures. And it really is a robust animal model if you know how to use it. There are some other C9orf72 models that give you maybe good pathology, but don't necessarily give you some of the behavior that you might be looking for. We have certain, theories upon that really kind of go back to health status. But for the most part, a lot of the transgenic animals are really good for DPR pathology and pathophysiology, but not necessarily so good for behavior. And this is a mouse model that we have really worked with to get not only the DPRs and the TDP43 with an antibody for TDP43 that actually detects phosphorylated TDP43, which is not the easiest thing to do as well. But kind of reduced it to practice. And so we've worked with a number of different clients who want to work with DPR therapeutics to use this mouse model to rapidly test their work. And so this is another area where I think we have a little bit more to do as a scientific community, is that the antibodies for TDP43, especially for phosphorylated TDP43, are really not very good. We're still using a polyclonal from the Mayo Clinic that we've licensed, but are frantically and furiously working to sequence and find other monoclonals that we can have an unlimited supply, and that works in both human and mice because we don't necessarily have one that works in both. And so this is just an indication at 6 months of age where you have repeats that are 149 repeats, and what the DPR or the TDP43 inclusion count looks like compared to 12 months of age. And so in this particular case, it's very progressive, it's very robust, and it's really reliable. And you can see this quantified here, as well as the immunohistochemistry shown on the right. And so, I didn't go through all the C9 models. I didn't go through the profilin data. I didn't go through the nuclear localization signals for TDP43 or the SING mice. These are all published. They're available, to look at PubMed, but I also have, data if you would want to look and see how we have validated these. But this is, some of the newer work that we're doing with Clotilde, who's here in the audience, as well as Don Cleveland. And again, we're really trying to make sure that these mouse models are available to the scientific community as quickly as possible. And so Kevin Egan has reported a Stathman-2 knockout with a humanized model. And these are available from the Jackson Laboratory. And we've made our own as well. And not only in addition to a knockout, but we've done, we've done them 9 ways till Tuesday, right? So we've humanized them. So if you're looking at therapeutics that are related to maybe antisense oligonucleotides or prime editing in those regions, we've done the humanization around that locus. We've also disrupted the TDP43 localization binding sites in this particular area. And this is an old slide, so I say if you look at our Stathman-2 models, you'll find that many of these are now available. We also have back transgenics as well, and I'm really happy to say that they're all without any licensing restrictions whatsoever. You want them, you order them. You don't even have to ask me for them. I won't even know if you ordered them or not. It's not a collaborative research proposal. It's just you order the mice and you get them. End of story. And so the other line of mouse models that we were looking for is a new back transgenic mouse model. This is maybe an instance of try again, but we really love Laura Raynham's model that has 500 repeats and has really nice DPR pathology. We have the one I made with Bob Balo that is unfortunately licensed under Cedars-Sinai. And we also have the one that we're currently making now. And I think when we're thinking about prime editing and different strategies where we're actually going in to disrupt, some of the repeats, we don't necessarily want lots of repeats and in tandem copies, right? So we want high expressing. And I think the other thing I'll say is that we have selectively bred all of these mouse models so that the repeat expansion keeps climbing. And so Laura's model we have at over 1,000 repeats now. So that's really great. But the repeat is different than the tandem transgene components to it. So if you are going to go in for prime editing, which I think is going to be a technology and a therapeutic that's going to be very interesting in the future, as well as antisense oligonucleotides, I think not having those confounding copy numbers is really going to be important. How am I doing? 2 minutes. Okay, so with 2 minutes left and right on time, thank you for the clock management, Hande. I hope I've recognized many of my collaborators over the years, and we're incredibly thankful, for so many members of the scientific community who have lent not only their brain power and their mouse models, but have been just terrific collaborators over the years so that we can provide this resource to the scientific community and hopefully expedite the work that we're doing in mouse models and ALS. And this is past and current members of my group, and I'm recognizing that the slide is also, somewhat outdated. But it really comes from a number of individuals who work as both lead scientists on the different mouse models and the genetic engineering, the science, the research, as well as so many people who perform the in vivo pharmacology and the clinical trial work. So, or the preclinical trial work. So with that, thank you for your time and thank you for the invite. Oh, you know what? And that's not Max and Max. That's Max and Laurent. That was, I know, I just realized that too. I'm like, okay, that's not the same person. But so Max Pressa is on the top end. That's Laurent Bogdanic, who's just an amazing scientist in the group. So don't want to not give him credit. So thank you.